Hello everyone and welcome to this webinar. My name is Adam Belaish and I will be sharing this session. So thank you very much for attending this webinar organized by MDDB and SECAM. For a small reminder, MDDB is a project aimed at creating an European database for molecular dynamics simulations. It's funded by the European Commission. Our main goal is to store and share molecular dynamics simulations respecting FAIR principles. Also, one of our goals is to provide talks, webinars, workshops, conferences for the community, and today's webinar is part of this initiative. Thus, we are pleased to welcome presentation by Dr. Yasaman Karami, Dr. Roy Gonzalez-Aleman, and Dr. Khakzad from the University of Lorraine in France. They are also great collaborators of MDDB, helping us grow and move the project forward. So thank you very much to them to present today. At the end of the presentation, you are all welcome to ask questions. And I won't say more and will now give the floor to them. So please, you can talk.
Thank you very much, Adam, and hello to everyone. So I'm just going to share my slides. Okay. Can you see my slide? Everything's okay?
Yes, we can see.
Okay, great, perfect. So first, I would like to thank you for this invitation to the webinar of MDDB. My name is Yasaman Karami. I'm a researcher at Inria Center and University of Lorraine in Nancy. And today I'm going to talk about DynaRepo, which is a database of MD simulation trajectories that is going to be integrated within the MDDB workflow and also the compass analysis. But also I have to say that this is a joint presentation, so I will present the first part of the work and then Roy will continue with the rest of it and Hamed would finish it. So before starting, I would like just to give you a very brief introduction. So biological molecules are building blocks of lives, and therefore it's really important to be able to understand their mechanism and their functional mechanism. So these molecules, they include proteins, RNA, DNA. And here what we see is an example of a mini cell. And if we focus, we can see that there are millions of such molecules. Therefore, it's really important to be able to understand the function of these molecules. But for that, what we need is to have access to their structures. And this is a great challenge. This has been a great challenge, especially experimentally and also computationally. But because of the recent advances of deep learning and AI, and thanks to the attention mechanism, there has been major improvements, and we had the outbreak of AlphaFold, which allowed us to have structures with very high accuracy, pretty similar to experimental results. And also this was reflected by the Nobel Prize in Chemistry that was awarded to the main developers of AlphaFold, along with David Baker for the protein design. So here we can just see an overview of the workflow. So I'm sure everybody is familiar, but what I want to say is that the success of AlphaFold owes very much to the development of AI and attention mechanism, but also to the data. This is the availability of data as a result of years of work from experimental and computational biologists, which allowed AlphaFold to reach to this success level. So if I want to sum up, from this introductory slide, there are three important messages. So here we are facing a data revolution. So the first point that we could focus on is the importance of data and how it played a crucial role for the success of this method in order to have accurate predictions. But this could be also applied to the other aspects. So for example, what we expect is to have another revolution, which is now focused on the dynamic, because we know that all these biological molecules, they're also highly flexible and they are flexible, they are dynamics. So now in order to be able to also understand the dynamical behavior, we need, and in order to improve the accuracy of all the predictions regarding conformational dynamics and functional mechanism of biological molecules, we need to have data under dynamical behavior. And this is one of the main reasons behind the efforts such as MDDB and why we also decided to work on the DynaRepo project. So there is a need for a collective effort, and we are trying to contribute to this collective effort. So this would be the first part of my talk. And another point, so the second point could be the importance of feature extraction and also pattern discovery. So once we have the data, the question is how to interpret this data, how to extract the most important data that is hidden in this data. So for that, I'm going to talk about Compass, which is a method that we developed within the team, and then Roy would talk about Intermap. And the next point is about designing novel architectures that would allow us to integrate this data for different downstream tasks. And Hamed would tell us more about this aspect of the study. So with this, I'm going to start talking about DynaRepo and to introduce what DynaRepo represents. So this is a repository of conformational dynamics. And for that, we teamed up with Hamed, Omid, and Emmanuel in order to build a database of conformational dynamics from a large set of macromolecular complexes. So we are mostly focused on protein-protein complexes, but we also have protein nucleic acid complexes, but also individual protein chains that I'm going to explain later. So in the workflow in general, what we do is first to select different important benchmarks, then perform MD simulation, and then apply the analysis and deposit the metadata and also the real data itself on the website. So this is a community-wide effort. So in fact, there are different people behind all the aspects, and this has been made possible also thanks to the help that we received from Inria Experimentation and Development Department, but also the support from MDDB, Gen C Supercomputing Center. So the data sets that we considered so far, because DynaRepo is a work in progress, so we are still continuing to work on this project. But so far, these are the data sets that we consider, and we performed MD simulations on all of them. So we have large set of protein-protein complexes from PDBind, from test 53, which is a benchmark of transient proteins. We also have a set of nucleosome complexes. We have a set of protein-protein complexes from ASBench, that is a database on allosteric. And finally, we also have a large set of antigens from SAPTAP database, the database of antibody-antigen complexes. So in total, we have something about 800 systems consisting of something more than 1,500 protein chains. So this was our data. And in order to obtain those data, of course, we applied several rounds of filtering, considering the resolution of these structures, sequence similarity, and many different parameters in order to end up with the input that we need for our MD simulations. So for the majority of the simulation, except the nucleosome, we use GROMAC software, Charm36M force field, and for each system, we perform 3 replicates of 500 nanosecond MD simulation. But for the nucleosome case, we use the Amber force field, NIMD as the software, and also more replicates and longer simulations. After that, what we do is to apply the workflow from MDDB in order to perform all the analysis. This would give us different information about the trajectories, such as RMST, RMSF, energy calculations, pockets, so many different type of data that we could extract. And we use all these data to build the final repository. And for the simulation, we've been awarded with 300,000 GPU hours from the grand challenge of Jean Zay using H100 GPUs. So this was an important step allowing us to perform this amount of large-scale simulations. So now what we are doing, so for the moment, simulations of the dataset that I showed you are over, and we are running the workflow in order to do all the analysis. This is also, we are half the way, so soon it will be also over, and then we will upload all the data to the site. So you can use these 2 links to have access to the node, to the Inria node of DynaRepo. And this is just an overview of what you would see. So we have the accession number of the simulation, here the PDB ID, and then these are the set of analysis that one could do. And then, so of course, there's visualization. So then you click and then you would see the visualization of all these features. And there is a specific tab to download all the data. So this was the first part of my talk. And now I would like to move on to the second part, which is about feature extraction and pattern discovery. So having access to these data to all the trajectories from MD simulation, now the question is how to analyze the data. And so why this is important? Because we know that, as I mentioned, all these biological molecules, they are flexible, they are dynamic, and also they interact together to form complexes. And majority of them, most of them, they exist as an ensemble of conformations rather than single frozen states. And also one important example where dynamic really matters is allosteric, which is a major obstacle that remains unsolved and is highly crucial for drug developments. So what does allosteric mean? It means that changes at one site are propagated across the structure to a distant site. And in fact, it's important to understand how such effects are propagated across the structure. So the goal here is really to understand communication networks within the protein that would allow the allosteric signaling to happen. So when we perform our MD simulations, what we obtain is a huge matrix that in fact represents coordinates of all the atoms in every step of the simulation. But now the main question is how to analyze such amount of data in order to extract pertinent biological information. And one strategy to do so is to use graph theoretic framework. So a few years ago, we've developed a method that is called comma or communication mapping, and this method allows us to start from an ensemble of conformations to extract a set of properties and then to be able to understand the communication network within the system of interest. So the properties that we extract are dynamical correlation, distances, interactions, and stability of secondary structures. And then from these properties, we define two notions: independent cliques that represent groups of local residues that represent high dynamical correlation and concerted atomic fluctuations. And a second notion, which is communication pathway. So here we have chains of residues that are highly connected together, meaning that they have strong non-covalent interaction and stable secondary structure and high communication propensity. Then we map these two notions on a graph, and from this graph, we extract communication blocks. So in a recent study with Emmanuel, we were interested to understand the effect of a post-translational modification on the nucleosome. So we studied the effects of cysteine hyperoxidation. So this is the place where we applied this modification. And then we performed large-scale molecular dynamic simulation of the Y type and also of the nucleosome with the modification. And then we use comma to be able to understand the impact of such modification on the dynamic of the system. So here what you observe is the difference between Y type and the S-sulfonylation. So in red, we have gain of pathway, and in blue, we have loss of pathway. So this point here is where the modification has been applied. And what we observe is that around the mutation point, there is a loss of pathway while we have gain of pathway across. So in fact, what we observe is that this modification brings destabilization of the DNA near the diet. And also it induces some rewiring of the local interaction network. But while this was really interesting, still we are missing some important information with respect to the DNA. So while this method was really useful, it wouldn't really allow us to go one step further to be able to understand the interaction between the protein and also the DNA. So with this, we decided to move on and to develop further. So we worked together. We built up a team with Sneha, Roy, and Emmanuel in order to develop a new approach that would allow us to understand and to extract communication networks within protein nucleic acid complexes. And this is how we develop COMPASS. So here we start from trajectories from MD simulation, and then we extract a set of features that are similar to before. So here we consider generalized correlation, interaction, communication propensity, and the distances. So then we put all this information together to build an adjacency matrix. And with this, we would build our graph. So this will be a weighted graph, and the weights are coming from properties that are extracted directly from the data. And the most important point in Compass is that to obtain this adjacency matrix, we are using PCA. So in fact, all the weights are assigned automatically. So there is no definition of cutoff. And in fact, this is an important point about Compass because it can be applied to any system. So in fact, it's a large scale method and it doesn't need manual modifications of the cutoffs and of the parameters according to a specific case study, but it's really a general method that could be directly applied to any system. And would allow us to build the network. So from this point, what we can do is to extract different kind of information. The first one is cliques, that are groups of fully connected residues. The other one is shortest pathways. So between every pair of residues, we use the Dijkstras algorithm in order to extract shortest and most efficient pathways. And then since we have a weighted graph, we can assign weights to these pathways and we rank them. And what we do is to provide the list of shortest pathway, like the topmost pathways. Then we also identify communities by using Leiden algorithm. And finally, we also propose a list of hotspots based on centrality measures. So here again, we have weights. So what we do is to propose a final list of top residues that are the hotspots of the system. So with all this, we went back to the nucleosome example, and now we applied Compass. So this is the results of Compass on the same study. Here we are looking only at the results from the wild type form. So you remember, this is the place where we applied the modification. But now this analysis allows us to understand how this position is connected to the DNA. So we can see that residues that are very close by are directly connected to the DNA. And this is how the effect of this modification is applied and how it brings the destabilization near the diet. Another example that I can show you is how we can use Compass in order to understand allosteric signaling within a given system. So here we studied the cysteine tRNA. So in cis-RS system, we have an active site and an amino acid recognition site, and we know that the two are allosterically coupled. So after performing MD simulations, we use Compass. And so what you see here are the top pathways identified by the method. And we can see that there is a nice pathway connecting the two sites. And this one is also going through the core domain. So this was really interesting for us. And finally, the last slide that I want to show you is the results on the allosteric hotspots. So here we consider 4 examples from the ASD database of allostery. So in yellow, we have all the hotspots from ASD, and in green, we have the overlaps between ASD and Compass. And also in red, we have all the hotspots identified by Compass. So the performance is not ideal, but still we have the results that are really encouraging, telling us that if we continue working on the project, we have a good chance of getting even better results. But for the moment, we can predict allosteric hotspots from almost 50% of the cases. So with this, I would like to give the floor to Roy to continue and tell us about another method for pattern discovery and feature extraction. Just stop sharing and let Roy to share his slides. Thank you.
use. And my name is Roy Gonzalez, and I will be talking about interaction fingerprints. As Yasaman just mentioned, this is a way of having meaningful insights from data. And when you have a lot of data, like in low molecular dynamics or molecular dynamics of systems that are huge, well, this is a fast way to have insights what's happening at the level of interaction between the two selection of your systems. Non-covalent interactions play a fundamental role in any biological processes. So we focus our attention on detecting some interaction like hydrogen bonds or van der Waals cell bridges in our system. And we like to keep track of where those interactions are established during the dynamics. And it is important to have a way to store and to compress the three-dimensional information. And one of those ways is by using interaction fingerprints, which have vector representations that contains as label two residues or two atoms of the system with what interaction is establishing between those and in which frames they get established. It's simple as that. But with that, you can, of course, make a lot of analysis regarding interaction prevalence or interaction lifetimes in order to see if it is a stable or is a transient interaction. You can filter docking results, but you can filter also your molecular dynamics by using some interactions that are meaningful for the system. You can cluster if you apply some similarity metrics those binary vectors, not RMSD, because we don't have three-dimensional coordinates in these cases, but you can cluster your different binding modes. And you can, of course, use it as an input for allosteric pathway detection, as just Yasaman exposed. And the problem is that for large-scale molecular ensemble, it is difficult to capture those interaction fingerprints, to detect them, to save them, and to profit from them later. I put just some systems that will get benchmarked later. I will show the numbers, but I put the number of atoms and the number of frames. These are not a super huge system, but even at that scale, they are considered very difficult to treat in the interaction fingerprints analysis. So we are trying to accelerate how we detect and save those interactions. From what exists in current literature, we can find that most of them are dedicated to document simulation, which is easier because we have not a lot of frames. And from those dedicated to molecular dynamics, we don't have a lot because from those four that we find in the literature that are the most used, only the two last one are able to process those dynamics that I just showed. And they struggle in terms of time complexity and space complexity of the algorithm that they use. The first time complexity bottleneck is because they have to compute a lot of distances very often. And then you have to compute angles because all those interactions are defined in terms of geometrical cutoffs and that include distances and angles. And they do so by using multiprocessing in Python, which can be slow. And the other bottlenecks regard the space complexity of those tasks because you have a system with a lot of coordinates and you have to load them in memory in order to analyze them. You have a lot of interacting pairs that you have to save in a way. And all those programs use uncompressed bit vectors. Well, they use byte vectors for real because in Python you don't have the data structure for real bits, and most of them use byte vector using 8 times more than what they should. What we did in order to skip those bottlenecks was to implement the distant computation step using KD trees, which is a data structure that allows us to decrease the complexity from n squared in the computation of distances to n log n in the average cases, which is very fast in comparison. We did also use Numba, which is a just-in-time compiler that gets a subset of Python instructions into machine code, accelerating a lot the computation of angles. And we use also Numba for the parallelization. the steps that are embarrassed in parallel, because we have a lot of frames, but each of them can be run in parallel without compromising the analysis. But we didn't use multiprocessing, which can be more slower. We use Numba for that. And in terms of space complexity, well, we cannot do a lot with the system coordinates because we have to load it also, and we have the same many interacting pairs. But we used compressed bit vectors, meaning that for each of those positions that are 0, 1, we use a bit, a real bit, and also the sparse vectors, meaning that you have a lot of zeros in your vector. We compress them to save some space, and we will see in a moment what are the impact of those design strategies into the performance of the tool. This is the overall workflow of Intermap. You have a stage where you prepare your system. You're just reading a configuration file, building the output hierarchy, parsing the topology and trajectory. And then we have the hard part, which is loading the coordinate in chunks and making the KD trees. These are time-consuming steps. Of course, the detection of the interaction is the most demanding step that we implemented all those strategies. And then we have to fill a binary container to save all that information and to exploit it later. And in a later stage, we save those to a result file with all the interaction fingerprints, or you can return a container if you're using Intermap as a library. You can return a container, which is a dictionary of the compressed vectors, and then you can pass to visualize. And before showing you the performance, I would like to build some confidence of the results that we are obtaining. So we compared Intermap, which is our alternative, with ProLift, which is the most stable, the most measured way to detect interaction fingerprints right now in the literature. So we have in the anion P, p-anion, and close contact cases that they are not implemented in ProLift, so they can be only detected by Intermap, but we implemented because close contact can be a way to see if two residues or two atoms are near in space, even if they are not interacting in the way that they are defined, the geometrical cutoff that are defined for each interactions. Then we have a zone that we have complete agreement between Intermap and ProLift. I mean, for cation P, p-cation, hydrophobic, van der Waals, anionic and cationic interaction, they are all detected in the same frames by ProLift and Intermap. And then we have some discrepancies between HB acceptor, HB donor. This correspond to the bug in ProLift that we are going to report because it's related to having the nitrogen of the peptide bond as acceptor, and they shouldn't be detected as such. And the other smaller difference are in the p stacking, but this is related to the way that Intermap gets the atoms, the rings, to compute the normal to the planes, and then compute the angles. I mean, these are two different algorithms in Intermap and ProLift. But when you visualize, you have the sense that there is no there are not chemically wrong. They are just different, detected in different frames, but they are both feasible. They are both physically plausible. So as the overall picture, we have that even if not identical, the results obtained by Intermap and ProLift are equivalent between them. The difference is that ProLift takes a lot of time in comparison with Intermap. You can see here the time we set a maximum time of 24 hours. And you see that ProLift couldn't complete the job for most of the trajectories that I just showed you in the first slides, only for the last one. And in that case, when you compare to Intermap, you have a huge speed up. And the other alternative is get contacts that have similar time performance than Intermap, even if, for example, for the last case, it's like 2 orders of magnitude slower. But we have to consider that get contacts is not computing the same of Intermap and ProLift. They are just reporting about 5% of the interaction. So in reality, this is an alternative that is not tailored for most of the system. And even if it is fast, it's fast because it's not computing the same amount of information. And in terms of space, meaning in terms of RAM consumption, we had a system with 64 gigas, sorry, is in gigas, the units. And we see that Prolif failed in several cases, just crashed without finishing the job. And for those that were finished, because we said one day as maximum, but in that day, they consume a lot more RAM than Intermap did. When we compare with Contas again, we have that Intermap is much more RAM conservative than the alternative, even when we know that Contact is doing a lot less job than us. And a part of the performance that is better with Intermap than with the alternative, we also propose Intervis, which is a visualization for visualizing all the data that you have in those iteration fingerprints. So it is a web application that can run without internet connection because it's not a web server, but once in your browser, where you can load all the results, all the iteration fingerprints, and you can load the topology of your system, and then you can select as much as like you do it in a VMD or using an MD analysis syntax, you can select which part of your system you want to visualize the interactions that you had. You can also select what interactions you want to visualize, the prevalence, you can set it just for not seeing the less prevalent interaction and not spending time in that. And then you will have an interactive plot that you can zoom, that you can save as a static picture if you want to publish, or you can save it as a dynamic, meaning an interactive plot to use it later. And you have this plot which show us the interaction between the selection 1 and the selection 2, but you also have a plot, two plots, one for selection, where you can see the prevalence of each selection that you detected in your whole molecular dynamics. You can also see the lifetimes of the interaction, because sometimes you have a high prevalence, but it doesn't mean that your interaction keeps all the time molecular dynamics on. It's just on and off, and then you can have this plot that will show you what is the lifetime of each interaction. And if you want to have the full picture, well, you can see the time series, which is the last tab of the application, where you can see the whole picture of what happened to each interaction system. Again, this is interactive, and you can focus your attention on what kind of interaction you want to see just by using the different options that you have in the page. So we hope that once published, it could be useful for users in general, and also for machine learning or other workflows that can use these interaction fingerprints and features and exploit them as a using all that data that is inside the molecular dynamics, but getting a nice overview of what is happening. So this is the part of Intermap, and I will let Ahmed to continue with the presentation.
All right, so my name is Ahmed Khakzad. I'm a junior professor at Inria Center, the University of Lorraine, and thanks a lot to MDDB organizers for having us to present our recent research works. So, Yasaman discussed about the importance of dynamic data and how we ran these large scale MD simulations to provide this data, and then also talked about the compass to extract meaningful patterns from the data. And then Roy also talked about Intermap. So now I would like to talk about kind of an end product. So how and how the best we can take advantage of having such data to develop systems that can learn the dynamic conformations of proteins or in general macromolecules. So I'm trying to go to, yeah. So the idea here was to basically build dynamic aware macromolecular systems, but of course this is a big basically goal. So we try to have a simpler task and basically design an architecture for the simpler task and then try to see how meaningful we can extract dynamic patterns. So this task is protein binding interface prediction. Basically, if you have a protein-protein interaction, finding the part and basically the residues that these two proteins interact through that would be the binding interface of these two proteins. Why it matters? Because basically it helps us to understand how macromolecules work and how they come together. Of course, through that, we can build a complex for the macromolecular system. And also knowing the binding interaction is of ultimate usage for developing novel therapeutics to target those interactions. But what is currently missing is basically capturing or learning the dynamic heterogeneity or conformation of macromolecules, in this case proteins. And also this would be important, for example, for addressing complexes that flexibility matters, like intrinsically disordered proteins, enzymes, or antibodies. To show the importance, I would like also to basically show you an example. Here you can see the two proteins interact that are intrinsically disordered proteins. So what we can see is this static interaction, and you can see in red the binding interface and in blue the rest of the protein. However, in reality, what we would witness is basically many conformations that the two proteins would have. And what we see is an ensemble of interactions between the two proteins. So using static approaches, we only see this a snapshot of the reality of the interaction. However, if we can understand and learn the dynamic heterogeneity of the two, so then we can predict the binding interface more accurately, and we can address the binding interface prediction when this flexibility matters and is important. So what about the state of the art? So of course, we have several approaches, mostly several deep learning approaches in the state of the art that address the protein binding interface prediction. Here, I focused and mentioned two most recent and important ones. So one of them is Massif. So Massif basically abstract the core of the proteins and focus on the surface. So what it does is to convert the surface of the proteins into a mesh and then extract meaningful geometric and chemical patterns of these surfaces, which is called fingerprints. And basically through a geometric deep learning, they learn important patterns that can be, of course, used for different downstream tasks. For example, they can be used for binding site prediction or ligand site prediction, or for search of interactors or etc. So the other approach is Pesto, which uses a graph transformer to encode proteins and in all atom version. And of course, Pesto is able to predict interaction site not only with other proteins but also with other macromolecules. So basically, Massif uses the surface information, ignoring the core of the protein. Pesto just don't discriminate between the surface and cores and just use all atom representation. And both of them basically consider fixed number of neighbors. And no, of course, obviously, they don't consider dynamic heterogeneity of the proteins. And so when we have flexible proteins, the accuracy reduce using the two approaches. So what we asked here was that if we can basically use both surface and core features of the proteins. However, we discriminate between them and discriminate between the importance of them. So with Omid, a PhD student in the lab, we considered and developed a type-wise message passing by using cooperative graph neural network. So just to give you a brief introduction. So basically, if we consider two nodes of I and J, so we have an edge connecting them together, and in a standard message passing in neural network, both nodes can send and receive messages. So this is the standard way of sending messages. However, in cooperative message passing, each node can basically have, of course, a standard message passing, but they can also only listen or only broadcast or be isolated. So this make a difference between the cooperativity in the graph neural network or the standard message passing in the neural network. So what we did, we differentiated between the core nodes, as you can see here in pink, and also surface nodes, you can see in blue. And so we had a graph transformer that encoded all the nodes, basically surface and core all together. However, to update the message passing, we used two cooperative graph neural networks, one for listening and the other one for broadcasting. And in this way, we developed a novel architecture that was able to basically regulate message passing based on the importance of the core and surface residues. So this architecture is called dynamic GT. So now I can give you maybe a little bit more details about this to make it clearer. Of course, it was trained over ND data that was come from DynaRepo. And so Yasaman explained already that this data is filtered from PDBind. However, as you may know, graph neural networks are data intensive. They requires lots of amount of data. So basically, we also use NMR and also AlphaFlow to generate extra dynamic data, basically covering the entire protein data bank for the training. So dynamic GT has two basically set of operations, the action operations and state operations. So here in the action operations, we have the two cooperative graph neural networks, as I mentioned. So one for the in logits or for listening, and the other for out logits or for broadcasting. So they regulate the message passing between basically all the nodes. However, then we have a mask state that distinguish between surface and nodes. So surface cores can have a standard message passing. Core and core nodes can have a standard message passing. However, between surface and core, we regulate this message passing and learn what is the best. And in the same time, this basically give a mask to our graph transformer. So in graph transformer, we have a key and query and attention mechanism. So we have our attention mask, and this basically action mask coming from the cooperative graph neural networks basically mask the surface and core residues and update the state operation based on the position of the nodes on the protein. And then we have the decoding part that or block that basically pull the informations from atom level to residue level and decode this information into predicted probabilities. So I mentioned dynamic features, so it's important maybe to talk a little bit about the dynamic features that we are using here alongside with the geometric features. So to define the dynamic features, I just want to give you a brief definition. Well, we consider X as the position of the atoms in conformation K as xk, and also we define movement vectors. So in a way that is the difference between the position of atom in the space, and then we normalize these movement vectors. So then we defined a couple of features for basically node features and edge features. So for node features, for example, we had root mean square fluctuation, which basically quantifies the average deviation of atoms from their mean position. And then for edge features, we had the communication propensity. So well, here we basically calculate the pairwise Euclidean distance between atoms, and then we calculate the variance of these distances in relative to the mean, basically position in all conformations. So basically, you need to consider each node having different positions in multiple conformations through MD simulations or through generative approaches. And then also to understand the movement of the atoms, we define two motion features, one of them called motion V and the other one called motion S. So basically, motion V is a cross product between motion vectors to see how correlated they are, and then to understand also the variance of this movement through the simulation, we also consider the cosine similarity of these motion vectors. And this is the motion S that shows basically the variance of the motion vectors alongside the simulations. So finally, you can also see here that if we basically record the listening and broadcasting of the nodes during the simulation, we can see that the network basically distinguish between the surface nodes and the core nodes, even through listening or broadcasting of the messages. So it shows that the importance of distinguishing basically between the position of the nodes on the structure, even for the message passing, is done well in the architecture. So for the performance, we went over a series of benchmarks. So I'm showing just part of them here. So you can just check the publication for to see all the benchmarks we did. So one of the perhaps most interested one was to use intrinsically disordered protein benchmarks. Basically all the benchmarks that are available. So we design different scenarios. Disordered binder, folding upon binding, and also basically mutual folding. So when both partners are disordered. So for each of the tests, we basically sampled the protein and gave the conformational ensemble to the model. The model basically gave us at the end the probabilities of each residue to be on the binding interface, and then we calculated the metrics. So here you can see the metrics over different benchmarks. So we had basically MFIB benchmark. We had a benchmark that recently published to compare, to basically show the performance of AlphaFold over disordered proteins. And we had the FuzzDB showing the fuzzy interactions in disordered proteins. And also we had the IDR-bind, which is another benchmark. So here also you can see that the metrics we reported, the MCC, the ROC AUC, and the PRS AUC. So showing that in most of the cases, we had an MCC, a high MCC, and also a very good ROC AUC for predicting the binding interfaces. All right. So we further also investigated how well we can predict interface residues on unbound states of proteins. So we went to ZDoc And so you can, as you know, that like not only gives us the unbounded states of the proteins, also it gives us some categories. So we were able to see different categories for the protein itself. So in most of the cases, again, our performance was high to predict the binding interface residues. Only for the case of antigens, it was not high as expected. It can be explained most likely because of the importance of dynamic in the antibodies itself, not perhaps in the antigen side. So maybe the dynamic is not that much important in this case, but this is something that needs definitely more investigation. So in conclusion, I would like just to mention that MD simulation data is gold standard and important. Why I'm saying that, you can see a simple test we did here. We only trained a subset of our model on MD data and then generated alpha flow for those MD data. So you can see there is a huge difference between the model performance if we had MD data and if we didn't have MD data. So MD really helped the model to generalize better and perform better. And so at the end also, basically building a dynamic ever model can mitigate the limitations that we have in the state of the art. And this can basically address flexibility in many downstream tasks. Here, it was just one example that nodes we use different listening and broadcasting, and it ended up to a different interface, to a more precise interface labeling. So it was a movie, so I couldn't play it, but well, we can go to our GitHub and see it. So yeah, with that said, I would like to acknowledge our collaborators for all these projects and also the team members, and to mention that we have open positions in our team. So please, if you found this researchers are interesting for you, so you can contact us. Thanks a lot, and we would be happy to take questions like this.
Okay. Thank you very much for these nice and really interesting talks. So we can now pass to the question sessions. So if you have any question, you can ask it by clicking on the Q&A button on Zoom. And in the meantime, I would like to ask a few questions. I have one for DynaRepo, Yasaman. So have you observed any patterns in allosteric hotspot or clicks, such as recurring type of residues or recurring hotspots regarding protein classes, for example?
So in fact, as I showed, we did for the moment, I mean, we are working on this part of the project, but we only consider few case studies. But for those, so we were really focused on complexes containing nucleic acids. And in fact, what we observed was that most of these allosteric hotspots are at the interface. So this was a pattern that we really observed from the analysis that we have done so far.
Okay, thank you. There is one question from Atakan Kubilay. So what's the typical time interval between different conformations, for example, from K to K plus 1? I guess it's for DynaRepo, the question, Atakan?
I think so. So for the simulation, except for the nucleosome, the interval is 100 picosecond. I think for the nucleosome, so it was Emmanuel who performed the simulation, but since there are longer simulation of few microseconds, the intervals are, I think it's 1 nanosecond for nucleosome.
Okay. In the meantime, I also have a question for Roy. And maybe I missed the information, but can you give me more details on how you compute the lifetime of an interaction?
Yes, sorry, I didn't put it an equation in the PowerPoint, but basically what we do is to see how much time that that iteration remains on your bit vector. And you have a value for that. Of course, if the iteration again established and gets stable for another part of your bit vector, well, you get that number again, and then you make a distribution, and you can see what are the lifetime, the distribution and the maximum lifetime that can help you to differentiate in cases where you have the prevalence, you have high prevalence, but lifetime that are not so high. So meaning that your iteration is just on and off, and is the transient iteration, or not super stable all the time of the simulation. And those cases where the prevalence may be not so high, but the lifetime is high, so it's an iteration that just appeared and get established for a period of time in your simulation.
Okay, thank you. We have another question. So, which package is used for simulation?
So again, for most of the simulation, we use GROMACS, as if by package you mean the software that we use, and Charm36 and ForceField. For the nucleosome, we use NAMD and Amber phosphate with, of course, modifications for the DNA.
I still have a few questions. It's now about dynamic GT. In one of your slides, you talk about node state, like listen node or standard node. I wonder how do you define this state? And like for DynaRepo, if you observe a correlation between the residue type and the node state, or the residue inside the node, I don't really know yet how it works.
Okay, so basically what we are doing so the residue type are encoded with within the network. So it's one of the features basically we are giving to the network. So residue type is basically there. So for the listening and broadcasting rate, so we are using a cooperative network. So we basically use the features that are there to update the messages between the nodes. So we can say that it's being updated automatically based on the position of the nodes, based on the features they have. So all the dynamic features or geometric features that I explained, they basically play a role to update this message passing. And this message passing would lead to the broadcasting and listening rate differentiation between the broadcasting and listening rate in different amino acids.
Okay, thank you. So there is another question for you. So from Adam Hospital, really nice example on how to use MD data to train AI models and predict dynamic flexibility features. How much do you think having a generic database of MD simulations such as MDDB can impact the field, opening possibilities for AI training?
Yeah, of course, it's extremely massive in my opinion, and that's also the main reason that we are really interested in what MDDB is doing, and we are also interested to help as much as we can, of course, and alongside the other researchers in the field. And as Yasaman framed it very nicely in the beginning, so I believe that the success of AlphaFold was not just because of the very amazing engineering that was behind it, but also because of many years of the data that was provided through the field. And now we basically see that the dynamic aspects of macromolecules are missing. So if we want to address that, we need a community effort to try to compensate such data. And I think MDDB is basically doing that, addressing that. And from our point of view, what we saw is that these dynamic features are really helpful and meaningful. So basically, we can see that if we train the network with data coming from stochastic approaches like AlphaFlow. So we get lower performance in comparison to MD simulations data. And this is just with the feature sets we define. So of course, if we go to to have more vectorial features and maybe to as time series networks, so we could even maybe see the impact more pronounced. So of course, the impact is huge for, in my opinion.
Thank you. So there is another question from FG Luke. Thank you for the presentation. Have you compared the signaling networks in proteins when distinct force fields have been used in the simulation?
So here we didn't really do that, but I mean, there's been many studies on that to see the effects of force field. So of course, Compass, like many other methods, it's not adding anything to what already is there. I mean, it really analyzes the simulation. So if you use force field X or if you use force field Y, and then you get two different trajectories. So when you apply Compass, it really tries to extract what is within the simulation. So of course, sometimes these differences could be important. For example, if you're working with disordered proteins, so for sure using charm or amber could have an important impact. So I'm sure if we compare, we would see different differences in the final network, but that is coming from the differences in the force field and the so of course it's also important if you have a simulation that has converged, if you have enough replicates, or if you just run a very short simulation with very few replicates. So all these things for sure can have impacts.
Thank you. Don't hesitate to write your question on the Q&A button tool panel, and I will relay them to the speakers. Okay, so I don't see any other questions, so I think we can close this webinar. Or do you have... Okay, I'll let you the floor.
Hi, everyone. We just wanted to bring your attention to the upcoming events organized by MDDB. We have two workshops before the end of the year, one in Italy from September 29 to October 1st. We have on-site attendance is limited, but there is also a broadcasting of the lectures. So you can register on SECAM website for this event. Same for the one in October in Lausanne. There is on-site attendance available as well as online attendance. And the final conference of the project will take place in Lausanne as well in February 26. These registrations are not open yet, but will be in the coming months. So if you keep an eye on the MDDB website and social media, you will see the announcement of the opening of the registration for the final conference as well. Thank you.
Thank you very much to everyone. So yes, don't hesitate to go on the MDDB website or on LinkedIn or anything. You can find a lot of information on the project. And I think, yes, we can close this webinar. Thank you very much to the speakers and to the participants of this webinar too.
Thank you very much for this invitation.
Thank you.
Bye.
Bye. Have a nice day.